Nanomedicine is getting smarter, improving our ability to treat cancer. The size of nanoparticles makes them ideal vehicles to carry drugs to tumors, and we can modify their surface and material to make them better at moving through the body to reach their targets. Our focus is on treating cancer stem cells, because if we don't get this right, then treatments might not tackle them, and these cancer stem cells will then lead to the recurrence of the tumor.
The goal is to transport therapeutic agents by using nanoparticles as carriers deep into a tumor in order to reach all cancer cells. However, most of those nanoparticles accumulate just at the surface of a tumor, not penetrating deep enough. There are two main reasons for that. The first one is dense extracellular matrix that hinders distribution of nanoparticles. The second reason is abnormal blood and lymphatic vessel architecture. It leads to reduced number of blood vessels, to irregular blood and lymphatic flow, and to high fluid pressure. Taken together, all of those aspects effectively block penetration of nanoparticles. To effectively account for those barriers, we need to very carefully design our nanocarriers. Therefore, in EVO-NANO project, we are using agent-based model of tumor growth, where we can very precisely represent spatial distribution and composition of all of those barriers in order to simulate how nanoparticles behave in such heterogeneous environment and how effectively they can diffuse in different scenarios.
We're using computer simulations to test how nanoparticle parameters impact their travel through tumour tissue. This includes things such as the size of the particle or how sticky they are, as well as how quickly they're internalized into the cell. We use a combination of agent-based models to work out how the particles travel through the entire tumour, to stochastic reaction diffusion systems to understand how particles move through subsections of the tissue. Right now we're interested in how space matters, that is, how tightly packed in the cells are, and the material around the cells and the impact of that as the particles move through. We're hoping that these computer simulations accelerates the design framework and allows us to work out which parameters matter the most.
One of the main appeals of gold nanoparticles is that we can attach a wide range of different molecules to them and use it to guide the nanoparticles to the preferred target. These attached molecules create a monolayer coating around the nanoparticle, which influences its behavior. Due to the size and physicochemical properties, functionalized nanoparticles can pass through the biological barriers and enter the cells. And the way by which they do this is really important because ultimately this is what determines their function. This process, however, is highly affected by the size, shape, and surface properties of the nanoparticles. So by understanding how it is affected, we can enhance cellular uptake. In EVO-NANO, we perform atomistic molecular dynamics simulations to investigate how the composition of this coating affects the structure in the biological solutions. And then, after obtaining the structure, we can move to a higher level, perform coarse-grained simulations, and investigate how these nanoparticles interact with cell membrane. Results provide guidelines to the more efficient nanoparticle design.
We are dealing with a wide range of scales, from atomistic to whole body scale. Different types of simulations are needed to evaluate at different scales the efficiency of drug delivery by designed nanoparticles. Executing these simulations require a large amount of computational power, but it's not only about how powerful of a computer we have access to. We are looking into finding intelligent solutions to predict the outcome of any given simulations at an earlier stage rather than waiting for the end of the simulations. Our vision is to use explainable AI to understand the intricate inner workings of the interactions of nanoparticles with the environment in which they release the drug they're carrying. We will further use this understanding to evaluate the efficiency of any given nanoparticle design.
We then use artificial evolution to optimize the nanoparticle designs automatically, so we can find new nanomedicine strategies with a click of a button.
By using a simple microfluidic model, we can apply gels that mimic human tissue and then also add nanoparticles that will mimic the nanocarriers of cancer drugs. We can then track these nanocarriers through fluorescent microscopy to gain an understanding of how they might penetrate tissue in the human body.
We are developing artificial tumor microvessel on a chip devices. These devices comprise of microchannel with a diameter in the range of the tumor capillaries and semicircular geometry. Also, they serve as scaffold of human endothelial cells to form an artificial vessel-like endothelium. The devices are used to investigate the transport characteristic of nanoparticle-based anti-cancer drugs in the bloodstream, mimicking as close as possible the human physiological conditions. The microvessel devices are also employed to study how nanoparticles of different sizes or chemistry interact with the vascular endothelium, and how these get extravasated out of the vascular flow to the surrounding tissue into the solid tumors.
The behavior of nanoparticles predicted in silico in EVO-NANO project are then contrasted experimentally. For this, we are using cancer stem cell model in which the cancer stem cells can be visualized and tracked. We are using 2D conventional cell culture as well as 3D models of the tumor spheres and in vivo models. Importantly, we can visualize and count cancer stem cells before, during and after the treatment. We could as well track its behavior since we know that tumor as such tend to compensate for the eliminated cancer stem cells in order to survive.
Our ultimate aim is to use our research to help patients. In order to do that, we need to translate our research results into a marketable product. Therefore, we are starting a new project that is financed by European Commission in the program called FET Innovation Launchpad, where we will first position intellectual property that stem from EVO-NANO in order to develop viable commercialization strategy and reach the market. We will do it through, of course, market research, through research of intellectual property in our domain, and through exploring potential service models, business models, and licensing mechanisms for various stakeholders in both academia and private sector.